hopefully I'm gonna walk everyone through, and hopefully I also don't talk too quickly and be knowledgeable of my audience. So like Giselle mentioned, so now I'm at the Technical University of Denmark, and I'm working in this Center of Excellence known as Chemist, the Center for Microbial Secondary Metabolites. And I'm going to explain a bit what we're doing here. So the outline of the talk is going to be focused on, so I've named it Enhancing Natural Products Discovery and Unraveling Chemical Ecology Using GNPS. So I'm going to walk through some of the work in my PhD that we published late last year, but also discuss and take some time to walk through the GNPS infrastructure. Of course, your facility there is quite used to all the tools in GNPS, but I figured it's worth walking through them anyway, but also walking through some of the newest tools inside. But first, we're going to start with Chemist. So what's the idea behind Chemist? So we, of course, as natural products researchers, we want to look for molecules produced from natural sources, right? And so obviously the foundation of natural products really is started with penicillin, right? Sorry, I might have dropped out of there. And so this discovery by Alexander Fleming kind of kickstarted, and then 100 years through research in 2019 when this was made, there were many more discoveries, and of course there were other problems like antimicrobial resistance starting and whatnot. But really the focus of natural products research and discovery was finding new antibiotics and how these benefit humans. The center here in Denmark is focused on why are these compounds produced? So why do the microbes produce secondary metabolites? Do they use them for antibiotics, or do they use them for other purposes beyond this? And what could their natural role in the environment be, and what at their natural concentrations? So of course, if a bacteria makes an antibiotic like streptomycin, it's not actually in that titer that you would prescribe to someone in the natural environment, we would imagine. And so this is the role of the center that I'm working in currently. And based on these principal questions, all of this kind of originates from the work done in John Clardy's lab at Harvard. And so, for instance, they just recently published this paper, but his lab has been working a long time on ants that cultivate fungal gardens. And so, for instance, this example that, again, they just published, there is on the left, so there's a pseudonocardia that is a symbiont with this type of ant. And of course, the ant brings back leafy material to a garden inside its nest, and a fungus breaks down this leafy material and the ants use it as food. Now, a fungal pathogen, Escovopsis, attacks this fungal garden and to try to kill it off. And the relationship that his lab has found is that this symbiotic bacteria, this pseudonocardia, produces a compound that inhibits the fungal pathogen that attacks the garden. So there's this really beautiful relationship of seeing the chemical ecology unravel through a single secondary metabolite and how this is influencing the overall system. So these are the golden examples that we can point to. And so in Chemist, we have three principal systems, all microbially based. So there's a marine system, of course, because here in Copenhagen, we reside on an island. And so we're surrounded by the sea. And so, and this is the expertise of the center leader, Lone Graham. So she's worked in the marine system for 30 or 40 years now. Additionally, there's a soil system, which I'm going to talk a little bit more about later. And lastly, there's a fungal system where we look at fungal rot with some penicillium species. So these are the three principal systems that we're working in. So we'll come back to one of the systems in a little bit, but let's jump into GNPS. So everyone hopefully here is familiar with GNPS. If we're not, it's basically this democratized platform to allow for originally molecular networking to occur. So molecular networking is a technique originating from 2012. And then this GNPS interface, this modern interface really starts from publication in 2016. And so molecular networking obviously allows for connections to be made in our data that might be more difficult to find just by looking at raw data. So for instance, the example they give in their paper is what happens is you are collecting Lcms data and two things occur to the data in the GNPS platform. So one, these MS spectra are queried against a library. So instead of a more traditional dereplication technique, where you're actually dereplicating information, so for instance, an exact mass, we're actually dereplicating using raw data. So it's a raw data match, and these tend to be more robust. They reduce the amount of potential isobars that you can come up with a hit in a database. But second, it links your data together within your file. So if you have MS spectra that are related, it will link these together and form molecular families. For instance, here they take one that has surfactant C14 and in the blue node, and there are two gray nodes. And when they dig deeper in the data, these gray nodes correspond to an extension or essentially a removal of a CH2 group. So we have these derivatives formed and we have a nice link. So, and if anyone's run molecular networking, we're all very familiar with seeing these molecular families. So this is the basic functionality in GNPS, and this is the tool that obviously is most widely used by the community. There's of course feature-based molecular networking. So this is a technique that relies on MS1 inputs to start making relationships. So the example by Luis Feliz is for bioactivity scoring. So in the publication, they take some plant extracts and they overlay quantitative bioassay data onto the molecular family. And here they make an assumption that peak area is related to bioactivity. So this is kind of in mass spec, this is kind of a dangerous assumption to make. But they back up their assumption by the fact that the larger the node, meaning the more peak area there is, the lower the MIC value of that compound is. So they show a really nice correlation to peak intensity, and versus the inverse is being MIC value. So this is a great functionality. And I'm going to show another example of feature-based molecular networking that is away from bioassay to show the meaningful nature of it. But let's keep going through the different tools. There's then Ms2 enhancements. So these are extra annotations that you can add to your data using some of these advanced tools. So one called MolNet Enhancer, and this allows for molecular formula. It takes predicted molecular formula and starts grouping them into molecular families. So you, for instance, the example here, you start seeing groups of triterpenoids or terpenes that have glycosides attached to them. And then you have a second tool called Ms2LDA, which kind of does fingerprint analysis. I'm going to go into this deeper in my talk, so we'll leave this here. But these are super useful to start looking at substructures within your data. Some of the newest tools in GNPS, which are worth mentioning. So this one's called Chemistry. It's a phylogeny-inspired enhancement tool. So for instance, on the left here, we have 3 Eppendorf tubes, and we can imagine they each have a sample from a different source. So let's say you have like bacteria, fungal, plant. Those are your three sources. Using kind of the ideas behind phylogeny and a root system on a tree, we can start seeing metabolites that are related to each other. So on the right-hand side, for instance, sucrose and fructose branch off at a different stage, and they're observed in different data files. And then you have this kind of weird phosphatidylcholine molecule that branches off very early and goes in a different direction. So the same ideas with evolution, or you're looking at 16S data, this is kind of a new way to visualize your data. Two of the tools that I'm very excited about and that I got a chance to be involved with a bit are these repository scale enhancement techniques. The first one is MAST. So MAST takes its inspiration from BLAST, NCBI BLAST, but instead of taking something like a protein, you are taking an MSMS spectra. So you enter an MSMS spectra and you query it into GNPS, and it will, first of all, try to match up and see if it does. So it creates a mirror spectrum like you see on the left, and can give you an identification of that query. But most importantly is it will return all of the data files that also have that MSMS spectrum. So for instance, this type of tool could be super useful. Say you isolate a metabolite from a bacterial culture. If you query this into MAST and it searches through GNPS's library and data files, you might start pulling out information on environmental samples that also see this MSMS spectrum. And you might start saying, okay, maybe my bacteria is in that environmental sample, and I can see the presence of that metabolite in that environment. So it's a super useful tool. The other tool is called Redo. So this tool uses a new metadata system. So it uses controlled vocabulary. You add metadata to your public samples, and then it allows for easy mining of this data. So you can pull out all of the bacterial samples, you can pull out all of the fungal samples you want, and then you can easily interface it and rerun molecular networking, or for instance, produce a principal component analysis plot like you see here. And for instance, the example here was looking at the presence of clindamycin, a broad spectrum antibiotic, in different human samples. So they have a data they query, and they look for clindamycin in blood samples, skin samples, etc. And so these tools are still, they require more input from the natural products community. There's a lot of information from medical samples, but we can imagine that as these tools have more data entered into them, they can start really revealing some meaningful questions on a wider base in terms of things like chemical ecology. You can relate, for instance, samples that you'd find in Costa Rica versus samples you'd find in another tropical location in across the world, or in other countries in Central America. So I think these are super exciting tools to use. So let's return to one of the systems in Chemist. So this is the soil system in Chemist. This is a park that just resides just outside of Copenhagen. Used to be the royal hunting grounds for the royal family. And so this was done prior to my arrival, but some selective isolation was done. We're looking for actinobacteria. So they isolated numerous species of Streptomyces, and we've gone through and they've sequenced these 6 so far. And one of the new PhD students in the center wanted to profile the metabolites and start seeing meaningful correlations. So I said to him, hey, let's do feature-based molecular networking. So we did OSMAT, or this is the one strain, many compounds approach, where if you take a bacteria and you grow it in multiple different media types, you see different metabolite profiles because you're inducing different biological machinery and hopefully in eliciting different compounds to be produced. Of course, we run this through Lcms MS. And so what does the data look like? So again, this was done by a student in the center. So here's the molecular network. Pretty complex. but let's just focus on a very small molecular family. So for instance, here's a molecular family. We don't know what the metabolites are. They're completely unknown. But the meaningful thing is Matisse here has overlaid multiple pieces of information, so multiple pieces of metadata on top of the molecular network. And he's made a nice legend for all of this. So the histogram on top are the different media types he's grown it in. So very meaningfully, you see that the feature at the bottom of the molecular family, 623, is produced much more extensively in mannitol soya media than the others, whereas the other two metabolites seem to be produced much more in a media type called ISP2. We also see that one of the outer circles is showing us which extract they're present in. So these metabolites are a majority in the methanol extract and a little bit in the ethyl acetate extract. And what's really nice is on the inside, he has created another pie chart which shows which strain is producing it. So very quickly, although it's a lot of data to look at, preliminarily, we get a lot of information overlaid onto one graph for prioritization. So we know, for instance, if we want to go after feature 623, we know we can probably culture strain P8A2, because that's the largest distribution in the pie graph, and also that we're going to produce it in potentially the mannitol soya media, if we want to just focus on that one media, on that one metabolite. So this is where feature-based molecular networking is, I really, really like it for type of strain prioritization. So whenever I'm doing some type of OSMAC approach or looking at it, I think this is super vital and a really great technique for evaluating these types of things. So yeah, that was, I think that's a really nice example of feature-based molecular networking. Obviously, we're going to keep digging through this data and hopefully find more meaningful relationships. We have some target metabolites we've chosen, but thankfully we have an idea where to target them. So the rest of my talk is going to be focused a lot on the work that was published in Molecular Omics here. This is open access, so thankfully anyone in the community can view it. But yeah, we wanted to first, Marcel, who spoke to you guys, might have mentioned before. So in the lab in Aberdeen, we're focusing on the extreme biospheres of the world. So in Marcel's lab, there are two systems that we typically look at, and those are the desert and the deep sea. So this project focused on the desert. So this is the Atacama Desert in Chile. And so the image on the left is more typically what we associate with the Atacama. So it's the driest desert in the world, and it's the Mars equivalent on Earth. So this is where NASA goes and sends rovers and things like this to be tested. And maybe surprising to some, not surprising so much for the microbial natural products community, is that, of course, there are bacteria in the soil, although very tiny amounts. Something that people that from the outside of Chile that wouldn't typically associate with the Atacama is the picture on the right. So there are these high altitude lakes that form. And of course, this is a super famous one. It's in lots of nature documentaries showing flamingo migrations. But something super important, and kind of going away from the idea that the tropics and the places of the biodiversity are the places we have to go to search for interesting compounds, is that even in these locations where we would assume life would struggle, there is a large amount of this biosynthetic dark matter, which has been coined. And so we really can go here and find potentially a lot of new biology, which we hope leads to new chemistry. So this is the main theme. So this project was brought to us by a group in Chile at University of Santiago. And so the student there went to the San Pedro de Atacama, and he focused on these lupine plants that flourish when rainfall drops over the mountains. And so what he wanted to do really was screen the rhizosphere, or the area around the soil, for Streptomyces strains that would have antifungal activity. The idea behind this is that Obviously, one of Chile's main agricultural exports is wine, and they have problems with fungal phytopathogens infecting wine-growing grapes. And so he did this screening procedure where he grew Streptomyces in the presence of fungi to preliminarily see antifungal activity. So he sent all of these strains in our direction to do the chemistry. So to start, we had some nice success. So here's a molecular network. I've removed all coloring to not make things complicated. So this is a more traditional approach. Drug discovery is the most frequently used example of molecular networking. So here's a molecular family, and in this molecular family, we have two known metabolites. So these are nystatin and amphotericin B. These are both antifungal polyene macrolides, and both are produced by Streptomyces species. Most importantly is the features in blue and the one in yellow are the ones of most interest. These are, so the ones in pink were grown from a standard strain that Diego sent to us, knowing that they produce nystatin. And I said to myself, we can network and see if it links to the known antifungals. So we have this metabolite 817. We did targeted isolation of it, and eventually it led us to this nice NMR spectrum, right? So using amphotericin B as our model, we can quickly scan through the Proton Nmr spectrum and say, what regions are we looking at? So of course we have an active aromatic region because we have lots of double bonds. We have these sp hybridized carbons that are attached to hydroxyl groups, so they're shifted. We have evidence of a sugar moiety in there, and then of course something very just easily to pick out is these methyl groups down at around 1 ppm. So in my mind, I'm thinking, great, we're on the right track. This is a metabolite we want to go after. The mass, when you dereplicate it and against the formula, looks completely new. So perfect, exactly what we want from drug discovery. Now, when we dig a little deeper into the genome, so using anti-SMASH and the whole genome sequence, we can start digging in. There were no gene clusters associated to nystatin. So this at first baffled me. And I said, what in the world is going on? So digging deeper into the type of metabolites that are associated to antifungal compounds produced by Streptomyces, we found this candidate gene cluster. It's a bit of a complicated mess, but essentially it leads to this type of gene cluster. Looks like it leads closely to a molecule that's present on the bottom here, this ECO 02310. So it's actually a linear polyketide-like molecule that we might be looking for. And so at first I said, why in the world is this linking to amphotericin and to nystatin? But there are a lot of the same moieties, and if we imagine fragmentation, it might be very similar. So if we actually take this linear molecule and look back at the same Proton Nmr spectrum, we again, if I highlight what we look at, we're looking at the exact same type of groups. It's just in a different layout, essentially. We still have all the same characteristics, and we still have the methyl groups we're looking for. So even though it's not exactly related to the molecules like amphotericin nystatin, this is actually a more exciting discovery than would have been just a derivative of one of those metabolites, because there are only maybe a handful of these linear polyketides. So a student is currently in Aberdeen trying to just get this in higher yields so we can get a nice, a really clean NMR spectrum. But it's a similar structure that we see to the one above. So the antifungal screening was working perfectly. But then we went on to another bacterium, so this one that we have pictured. So this is Streptomyces S29. And to kind of mimic what we're seeing on the plate so that we can scale up appropriately, we wanted to do this on liquid. So we did this in a 10-to-1 ratio of Streptomyces to fungal culture. We did this with two different pathogenic fungi, phytopathogens, Aspergillus niger and Botrytis cinerea. And then we did typical natural product screening. So we went through NMR and LCMS, looking for antifungals like before. But we didn't see anything. No antifungal metabolites were present. Nothing according to the gene clusters that we saw or anything. But super interestingly, when we streaked the end culture, we saw no presence of the fungi. So we knew that the Streptomyces was doing something to prevent the fungus from grabbing hold in the liquid culture. Now, of course, we've given the Streptomyces a really overwhelming advantage. And we want the Streptomyces to win, but still the fungus has mechanisms for combating it. And we would expect the Streptomyces to produce an antifungal compound to kill it off. But actually what it turns out to do is it produces Sedarapores in order to essentially starve the fungus. So what are Sedarapores? Sedarapores are iron carriers. So these are the 4 principal types of microbial Sedarapores. We're not going to go into all of them, but essentially Streptomyces and other actinobacteria are really good at making this hydroxamate type of Sedarapore. They almost form this peptide backbone. And in talking about Sedarapores, there are these 4 what they call social interactions. So the first interaction is that you can imagine you have 2 of the same bacteria. They make the same Sedarapores, and so they can share Sedarapores between each other. And they're perfectly happy doing this. So 2 Streptomyces species can make the same Sedarapores, and they can share them amongst each other. And there's no problem, tends to be no problem with this. The second interaction, which is cheating, is a much trickier one. So say you have 2 bacteria, one produces Sedarapores, but then the other one has the uptake mechanism to steal those Sedarapores away. I think this mechanism is also known as piracy. So we can think of it this way. And so this is kind of what they see in cystic fibrosis patients when they're given iron supplements. It actually promotes the growth of certain pseudomonads because they can uptake the iron from some of the Sedarapores produced. So it actually makes infections worse. D is kind of a complicated competition mechanism, but C is the one we want to focus on. So C is what we believe we're seeing in our system, and you'll see that as we go. So it's a locking away mechanism. So essentially we can imagine that the Streptomyces is the green organism. It's producing Sedarapores, and then it can go out and take iron and bring it back into the system. Whereas the fungus is the purple organism, and it neither has, so it makes its own Sedarapores, of course, but it doesn't have the uptake mechanism to take the Sedarapores from the Streptomyces. So essentially the Streptomyces can just steal the iron away, and then the fungus can't actually grab a hold. So how do we know we're actually seeing this, right? Or we'll get to it. So in Streptomyces, the hydroxamate type of Sedarapores are called desferrioxamines. And essentially they have a so-called life cycle, which I think helps explain them. So of course, in the bacteria, you have metabolite pools, which are full of primary metabolites. And here we have a very simplified gene cluster that produces desferrioxamines. So these metabolite pools can feed the gene cluster. And so in the presence of lysine, it feeds the first gene in the gene cluster, and that converts lysine into cadaverine and further on into the workflow. But if there's a lack of lysine, this first gene or first protein can actually be bypassed. So if there's enough putrescine in the environment, it can just bypass the first gene and continue making them. But of course, every molecule is now made with putrescine instead. The most promiscuous of all of the enzymes is this third one, DESC. Essentially, it can bind any type of organic acid that can similarly bind something like acyl CoA, right? And so it can bind all these different organic acids, and essentially it pieces these molecules together in a oligomer fashion. Once all of these are made, of course, it's exported, it chelates to iron, and then it's imported again. So this is the life cycle of these metabolites. So we did all these co-cultures. we did monocultures as well to ensure that we had nice data to replicate against. And we ran a molecular network. So this is a very small, this is a molecular family, the biggest one in the larger molecular network. So here, I hope the main takeaways here are a couple things. One, so we had super great anchor points from the GNPS library because these are very well-known metabolites and they've been deposited and studied multiple times. So any hexagon is a known desferoxamine identified in the GNPS library. Every square is a newly discovered, newly annotated analog of desferoxamine by myself. So you can see there's lots of them. And then every circle that's left is an unknown. They're connected through the molecular network. So we would assume they are desferoxamines, but they kind of have weird molecular formula. We can't explain away what's going on. And then I've color coded them according to their structural diversity. So we have kind of this blue color. We have this N-acetyl cadaverine moiety at the terminus, and then we have this desferoxamine D-like metabolite that has an acetyl group at the terminus. And then we have this purple metabolites that have, well, in this case, phenylacetic acid moiety, but they can have any type of aromatic ring structure. So we see a lot of this, and there's a couple molecular families, but essentially I thought it was worth going through and discussing how the annotations occurred and knowing having some assurance of what we're seeing. So for instance, let's take an example. So if we look in the molecular family and we pick out the feature 673. So here we've run all of our data through an accurate mass instrument, a Bruker Maxis. And so we can predict the molecular formula. And it just so happens that the one that has 100% score is the correct molecular formula. This doesn't always need to be the case, but we also have a good idea what desferoxamines, their formulas should look like. Of course, we've collected MS data, so we have a fragmentation pattern. And then using the fragmentation pattern and the molecular formula, we start piecing together substructures. So for instance, from the fragmentation, we can piece together this larger substructure that accounts for a majority of the metabolite. And then we essentially piece together what could be left over. So we have these branched acyl chains down here in the second part of C. And obviously, when we come to the end and we have a potential putative structure, we want to go back and confirm. And thankfully, we have accurate mass fragments as well. So we can fragment our molecule in ChemDraw or a similar software and go back to the MS data and try to confirm if these line up properly. So we see really nice information. Something super useful is that these metabolites are peptides, so they fragment in a uniform way. When you're talking about other metabolites like alkaloids, flavonoids, things like this, fragmentation's a little more tricky to predict. And so it can be more problematic for trying to come up with new annotations for those types of metabolites. But in this case, for peptides, it works really, really well. We, of course, see a molecular family or multiple that have chelated metals still attached to them. So these are chelated features. So these are features that actually have associated features that are unchelated. So again, the hexagons are known desferoxamines deposited in GNPS. And a seemingly an unintended consequence of being able to chelate iron for these metabolites are that they also can chelate aluminum. So iron is an essential metal for producing cell wall and for other functions in the organism, but aluminum is actually a toxic metal. So it seems to be completely unintended. The organism, so far as we know, doesn't use aluminum. Somehow it must be getting rid of it so it doesn't kill itself. But sometimes we see these metabolites hold on to the chelated metals. Typically, you assume that they lose it through chromatography, but in this case, we still see them. So we have really nice confirmation that some of the new features that we have annotated, they have correlated, chelated features that can back up our annotations. And so we have quite a wide structural diversity. We have these purple aryl desferoxamines, and this leganoxamine A glycoside is the second desferoxamine with the sugar moiety attached to it. So it's a super interesting metabolite. It seems to be that the ability for some type of post-translational modification to occur, some glycosylation is present there. And then we have these kind of truncated desferoxamine-like metabolites, the tenacibactins. They have an acyl chain off the side, but they're a little shorter. And they have a succinate moiety on the C-terminus. And we have these desferoxamine D-like metabolites, which I think our publication is the first to describe these derivatives. And then we have some kind of more odd acylated desferoxamines. We have an unsaturated one, which they've been described before. We have these really, really odd dicarboxylic acid desferoxamines, which hadn't been showed before. So the one caveat with these types of metabolites is that they are really difficult to get in a high enough quantity to do NMR studies. So annotations in this study were strictly based off MSMS. So in that case, we have to assign confidence for these. So there's a couple different ways to do this, but in my opinion, Szymanski's rules are one of the best tools. So this is a figure, I'm sorry, it's a little blurry, but we can still see the main points. So Szymanski's rules identify feature confidence with 5 levels, where you can see 1 is the top level, where you've confirmed the structure by using a reference compound, and then you have additional experimental data on top of it, and 5 being the worst. So you have an exact mass and maybe a predicted formula. And we want to be, of course, we want level 1, but for us working on potentially new compounds, it's very difficult to have a reference standard for something that is brand new. So typically we are working in the ranges of level 2 to 5. And ideally, we want to aim for 3 is really a nice level. And using GNPS as a tool, this is highly possible because we are actually comparing MS data and linking nodes together through the experimental data. So in my opinion, level 3 is the starting point for GNPS. The majority of the metabolites in the study here were level 2 and level 3. Level 2, based on the fact that they had library matches in GNPS, and level 3, based on the fact that they might be connected to those anchor points that I was mentioning. So the overall question is, and why we saw no fungal growth, is what's the chemical ecology that we're seeing actually happening? Does co-cultivation elicit a response from the Streptomyces? So when we grow Streptomyces alone in iron deficient medium to induce production of the Sedarapores, we see 24 new analogs and 8 known analogs. So from the start, we already know that our Streptomyces makes a lot of new diversity of Sedarapores. So it's a very unique Streptomyces in this case. But when we grow it with a fungus, and at the end of the day, it turned out not to matter which fungus we grew it with. We saw the same exact response. We see an additional 22 new analogs on top of it, an additional 17 known analogs. So we see almost a 50%, well, we see a 100% increase in new metabolites induced by the fungus being grown with the Streptomyces. So we're really seeing a nice ecological response by the organism. And really tying into everything is that the majority of the metabolites we see in the co-culture were acyl desferoxamines. And these were shown by Matt Traxler and Peter Dorstein and Roberta Coulter's lab in Harvard. that these metabolites are produced almost exclusively when grown in actinobacteria co-cultures. And so potentially we're also seeing the same interaction happening with a Streptomyces and a fungus that they were seeing with the 2 Streptomyces species. And so potentially these have a larger ecological role in locking away iron or having a competitive edge over other bacteria. So this is a really nice tie-in to previous ecological studies on these metabolites. So kind of slightly leaving the ecological side and thinking about the overall chemistry is, have we tapped out the chemical space? So desferoxamines have been studied for a long time, and they're very well characterized metabolites. And seemingly, in my study, I find that discovering a lot of analogs, but it's kind of difficult. Not very many new derivatives or new analogs, new structures are found every year. So using this tool that I mentioned earlier, Ms2 LDA, we can start looking at these relationships. So Ms2 LDA, the idea behind it is that each metabolite leaves a different fingerprint, Ms spectrum. So for instance, we have 2 different metabolites here, ferulic acid and ethylphenol. And you can see their fingerprint spectra in red or blue. And when these metabolites are essentially pieced together, we can still see evidence of the components making them up in the fingerprint. So the blue matches up in the smaller component to the larger metabolite, and so on and so forth. So this idea is that we can take these fingerprints and start coming up and inducing or start finding relationships in these fingerprints. So how does this look like with these desferoxamine metabolites? So here on the left-hand side, we have 2 desferoxamines. They just differ in a CH2 group on the acyl chain on the left-hand side. And on the right, we have their MS spectrum. So in this tool, you go in and you annotate your own motif. So for instance, I'm going to annotate a motif now. So these structures fragment in the exact same place and produce the exact same mass. So this is part of the characteristic motif of these desferoxamine structures. So they produce these 2, but then they also take into effect a similar loss. So both of these metabolites lose 160 from the N terminus, even though the masses don't line up perfectly. So at the top molecule, you have 499, on the bottom molecule, 513. All this information gets tied in and we say, this is one Ms2 motif. And so we can use this information and start maybe finding these fingerprints in other features that we wouldn't normally associate to desferoxamines, because maybe we don't have an anchor point or we don't know that we're supposed to be looking in that area. So when we run Ms2 LDA, and I suggest to anyone, the documentation's really good on this. Also, the developer of the tool, Justin Vanderhooft, he's really lovely about helping out. So if you send questions to him, he's happy to help walk through things with you and do all this. So here on the left-hand side, I've overlaid, so this is the original molecular network with Ms2 LDA overlaid on top of it. And I have highlighted all of the nodes that were annotated in the original molecular networking job. So there's 107 nodes, 107 features, and they correlate to about 60 metabolites. And they have 970 edges, connections, right? So what happens when I select for all of the Ms2 motifs that I have curated myself? What happens is we see about a 2.5 times increase in the amount of nodes and a doubling in the amount of edges. So we see a lot more chemical space that's out there. Super exciting things is we see a ton of single nodes that aren't linked to anything else, that look like they're very unique chemistry. But also, if you look at the top row, the 3rd molecular family from the left, in the original molecular network, it has absolutely nothing highlighted. But in the overlay network on the right, it has its whole network highlighted. So clearly this is one whole molecular family that is full of desferoxamine fragmentation fingerprints, but we don't have any idea of any of the metabolites inside of this molecular family. So this is somewhere to target discovery. So overall, in this study, so we took something that we wanted to find antifungal metabolites, and the original goal was not chemical ecology yet. But through looking at GNPS data and evaluating the mass spec data, we saw this explosion of desferoxamines, and potentially that pointing to the fact that the fungus didn't survive and was starved of iron. And then using MS2LDA, another tool in GNPS, we can actually see that the chemical space has a lot of room for expansion in these metabolites. But ultimately, at the end of the day, what's important is it could potentially lead to the same end product that we have. So this organism from the rhizosphere of this lupine plant, it has a Streptomyces that has some way to prevent a fungus from grabbing an ecological niche. And ultimately, we wanted an organism that could protect a plant from fungal phytopathogens. And it looks like, although this doesn't produce a single compound to knock out a phytopathogen, it uses a different mechanism to try to protect and prevent anything from grabbing that niche. So it's still a really nice story that we have behind it. And so lastly, I just need to thank everyone. So we need to thank, obviously, Chemist and the Danish National Research Foundation, who's funding my work now in DTU and further chemical ecology studies. And there on the right is the group in Aberdeen. And so thanks to everyone there, especially the master's student, MT, who did a lot of the experimental work for this while I was writing my dissertation. And she got very minimal feedback. She was an excellent student. And the group in Chile at CBIB, the Center for Biotechnology and Bioengineering. And lastly, thank you all for listening. And I'm happy to take any questions.
Thanks, Scott, for this tremendous talk and looking into these incredible tools that you have to use. I have a question here from Daniel. Did you try any kind of chemical elicitation besides from co-culture?
So, we didn't use any other chemical elicitations. We didn't add any other metabolites like antibiotics, but inherently the medium type that we typically grow the organisms on, ISP2, is an iron deficient medium. So although we haven't added a metabolite, we've actually removed iron from the environment. So in our normal cultures, we are still eliciting a response by removal of a metabolite. But no, we didn't try anything like, typical like kenomycin addition or anything like this. Be interesting to see if we maybe threw some like fungal produced antibiotics at it, maybe what the Streptomyces would do then. But yeah, definitely something maybe someone should follow up on.
Thanks, Scott. There's another question. Did you continue working with the long chain polypeptide that was being produced on Petri dish, but not on liquid culture?
So say again, I'm sorry.
Did you continue working with the long chain polypeptide that was being produced on Petri dish, but not on liquid culture?
Yeah, so so there's a PhD student who is trying to grow it in a much larger scale to produce it. And at the same time, the group in Chile so they've developed a new technique for producing a mutant to transfer this really large gene cluster, which is like 200,000 base pairs. They're going to transfer it into a heterologous host to try to, one, do mutant studies to look at the biosynthesis, but also to try to produce a promoter strain so that we can get really high yields of it. But we have tied the mass to the antifungal activity. So we've isolated, the student at Aberdeen has isolated a fraction, although it was only a very small amount of the metabolite, but it's the principal component, the major compound, and it is absolutely the responsible metabolite for the antifungal. So there are people continuing working on it.
I have a question here, but before asking that other question, did you actually harness another actinobacteria with potential antifungal activity besides these Streptomyces?
So the group in Chile is strictly interested in Streptomyces. And so that's what they sent us. But this is something that I am hoping to set up in Chemist. So I have started doing isolations of rare actinobacteria because I think, yeah, there's a lot of chemical space to be explored there, and they're also present in the soil, not just Streptomyces. Of course, the community is very interested in Streptomyces because it's the, you know, #1 antibiotic producer and all these things. But yeah.
Mm-hmm. And because the question that was posed here is, if you study as well the chemistry of the pathogens of the, if they are producing something that are controlling the Streptomyces as well.
Yeah. So we didn't. So initial experiments. So I had to learn a bit how to. So in the beginning, we wanted the experiment only to evaluate the Streptomyces response. But in the process of going through it, of course, we learned the lesson that it's very difficult to work with an actinobacteria and another fast-growing organism. So if you want the actinobacteria to ever survive, you have to really pre-grow it a lot, or else the fungus just grows so quickly that it will overtake it every time. So we never got far enough into the stage of then doing the inverse relationship. But of course, I would imagine that if you grow the fungus, so yeah, it's difficult to balance the fact of getting the fungus to produce its secondary metabolites in order to overcome the Streptomyces, because it essentially just tries to outgrow it if you grow them at the same time. So these experiments are a little difficult to do, but that's something that we're looking at here in Chemist as well, is that we don't want to just look at the one-way relationship. We want to look at both directions. And that's where techniques like imaging mass spec really come into handy and start allowing for those types of relationships to be seen in a nice way.
Mm-hmm. The plant that you actually choose, I mean, that was chosen by the Chileans, right?
Yeah. Yeah.
How was it chosen? I mean, it was because it never showed a fungal infection, or?
Yeah, so it was chosen for, well, I think it was chosen mostly for the reason that it survives in this high altitude, high UV, low rainfall, high salinity environment, but also the fact that these lupine plants, so they remain dormant, the seeds remain dormant for many, many years. But I think some studies have shown that the soil bacteria that exist around the seed actually somewhat mimic the environment when the plant grows. So they believe that the rhizosphere bacteria actually protect the seed while it's lying in wait for enough water to germinate it.
You know,
yeah.
Judging from your experience that when you have growing both organisms, the fungus actually overcome any potential growth of the bacteria. How does actually works in the environment? I mean,
yeah, it's a good question. Is it just maybe there's there's a lot more of a bacterial load, or are there, of course, there are other fast-growing bacteria, so maybe they steal up the ecological niche faster than the fungi can, because there's pseudomonads that grow very quickly. So it's this is something that's very difficult. I know the center here is very interested. We have a lot of work on making synthetic communities with 3 or 4 different bacteria and seeing how different secondary metabolites affect the dynamics of these communities. But it's very difficult because you imagine in the soil, in the lab, we're not mimicking this environment in any way. In the soil, you have a bacteria over here, and you have the same bacteria all the way over here, and then you have millions of others in between communicating. So this is the great mystery, right? What's actually happening in the soil?
Mm-hmm. There is a question here, but I don't know if actually, I have a question related with the data treatment prior to FBMN. Do you usually perform blind signal subtraction prior to MZ-MIND processing, or do you subtract the blind features after MZ-MIND processing?
Yep. So I actually subtract. So if you do like chromatogram subtraction in a processing software, it can actually kind of mess with the way that your features are pulled out. So if I'm using MZ-MIND, I actually run the blanks alongside the process, and then at the end, what I actually do is when I export the CSV file, the final file with alignment, I then in Excel sort the data by the blank, and I will remove any feature that contains a certain peak area that's in the blank. I'll remove all of those files, all those features, and then I'll send that final CSV file to feature-based molecular networking.
Mm-hmm. Okay.
And then I know in MS-DIAL, they have a parameter where you can select, so you identify what are blanks, you select you want to remove them, and then you can do you can do a removal step in MS-DIAL. Yeah.
But you prefer to use MS-DIAL instead of MZ-MIND?
So I think they both have advantages and disadvantages. MS-DIAL is much more simplified, and so I think for beginning users, it's very easy and it's very fast to learn, but it is lacking some major features. So for instance, you get a lot more in MS-DIAL, you find a lot of features pertaining to isotopes, whereas in MZ-MIND, you can filter out the isotopes. And so this is one caveat. So MZ-MIND, I think, is a more technical tool, but it has more ability to pull out that information you don't want. Mm-hmm. I believe MZ-MIND is producing a new workflow, so MZ-MIND 3. And my guess is that they're going to make a more streamlined approach that like MS-DIAL contains. So the hope is that MZ-MIND has a much easier interface, but still with all of the components that makes it maybe a better processing software at the moment. Yeah.
Okay. So I don't see any more questions here. I put in the chat the reference of Szymanski for the people here interested in knowing the level of confidence that you can report compounds, identify compounds. I don't know if you know of another which was launched, I think it was last year, which incorporates data from other spectrometries as well, NMR and IR and UV. And then the level of confidence is with letters A to, I don't know, G or something like that. I don't know if you were aware of that.
No, I didn't know that.
Well, I think, I mean, they have to publish it. I think it was put into yeah, I mean, not a preprint. It was a consultation given by several laboratories if they will be willing to embrace this other level of confidence as well.
Yeah, it's always a challenge, and we've spoken about this. It's always a challenge having, is your feature, how true is your feature?
Mm-hmm.
It is always a difficult thing to say. I think the really nice thing about having GNPS giving you information on this is that, so of course, if you're looking for unknown unknowns where you have no reference point, it's very difficult. Although you have the information of MS data linking to each other to help confirm and using this experimental data. But sometimes I always go back to when I was trained as an undergraduate and like going back to basics, like isotope distributions.
Mm-hmm.
If you don't see the C13 isotope distribution, you can't call it a peak.
Mm-hmm.
And these simple things. And the good thing is if a peak, if a metabolite or a feature is selected for MS, you know it reaches a certain threshold, and you would imagine that it is having a correct isotope distribution. But it's always important. So for instance, all of the annotated structures that I've done in the work I did, I went back to the raw data and confirmed that these look like real features. And now GNPS has a really nice new function called Lcms Dashboard. And you can go in and you can visualize all this data. And here, I'll make sure I'll put it into the chat. But it's a really nice way to, so you can convert your data to MZML, and you can place the data into this. And essentially it's like a analysis software to some degree, but they are working on interfacing it so that you can instantly go to molecular networking, or you can instantly do feature finding in a very crude way and send it to feature-based molecular networking. Mm-hmm. So for instance, this tool is super useful, and they've shown on Twitter. So for instance, they took my publication and they looked at the public data and they said, okay, it's basically is Scott telling what he's saying, telling the truth, what he's saying in the paper. So they found the feature, they looked in all the data files, they can clearly see a nice extracted ion chromatogram in only one file. And so these types of things instantly show if a feature might be real or not, or what it looks like in the data. So yeah.
Okay, no, great. Well, Scott, thanks very much for being with us today. It was a very interesting talk, and I think it's more related to the work that Ciprona is making right now. I mean, looking into chemical ecology, I think it is the right path for future natural process chemistry in terms of the resources that we have here in Costa Rica. So we will be quite open with potential collaboration with Chemist, and yeah, I mean, we are open to discussion.
Absolutely.
And I think,
okay. Thank you all. Have a nice day.
Have a nice day, you too.